Hopefully you all saw on the website, we've created this Oracle VirtualBox OVA file that has everything pre-installed on it, all the software and the tutorial files. So basically it's a little version of Unix that can run inside your existing operating system, whether it's Windows or whatever. So if you haven't done so already, if you could download the VirtualBox program and then this OVA file, that'll help a lot. And really, if you just go to the last link there, that'll take you to instructions on where to download, and that's probably better than visiting the first two links. So I'll give just a second to write down the last link. And you know, if you want to just watch me do it on the screen, you could, but it's nice to be able to follow along. So the first section then is going to be about this program called POVME. This is a big review because this morning you guys just went over docking, but POVME is a program that's useful for picking out receptor conformations that you could use in a virtual screen. So there's a quick overview. The way I see docking, I see it as having two steps. First step is you take your small molecule and you position it within your binding pocket. And then the second step is, once it's positioned, you score it with a scoring function which tries to map that binding geometry to a correlated affinity. So it's super important when you're doing docking then to pick the proper receptor structure. And it's amazing how you can get very, very different results depending on which structure you use, because you know, these proteins, they're not just, static structures, kind of crystal structures, lie to us because they give this impression that the protein is just a single conformation. But really, these proteins are breathing in solution, and the conformations of those binding pockets are always changing. As an example, this is some work I did a few years ago on a protein called REL1 from Trypanosoma brucei, which causes African sleeping sickness. What I want to call your attention is that residue E60. So I have one protein conformation shown in that sort of glossy solid representation. And you can see there that E60 and R111 are pretty far apart, such that there's this little sort of crevice in between them. And so we did some virtual screening into that conformation, and we identified the compound you see there, which was subsequently shown to actually inhibit the protein in experiment. But if you look at the, and I hope you can see it, the mesh representation, that's another conformation of the same protein. And in that particular conformation, that E60 is moved right next to R111, and there is no cleft. So if we just looked at one protein conformation, this pose would not have bound in the way that we presume is the correct pose. We don't know for sure. And we wouldn't have identified this molecule at all. So it's very important to consider different conformations. And there's many different sources. One good source is just to look at crystal structures. Oftentimes when you co-crystallize a protein with different ligands, you'll see different conformations that way. But we really like, in the Amaro lab, we like molecular dynamics simulations as a way of generating many, many different protein conformations as well. There are thousands and thousands of them depending on how long your simulation is. And then the question becomes how do you pick out notable conformations from the simulations to use in screening, because to dock into thousands would just be impractical. And that's really where POVME can be helpful for finding these sort of notable pockets among the many thousands produced by an MD simulation. So, you know, some of the questions you might wonder is, over the course of this whole simulation, what's the biggest my pocket ever gets? Or what's the smallest? Or are there any of these little side pockets, as we saw with REL1, that might open up that I could exploit pharmacologically? And so what POVME does, POVME stands for Pocket Volume Measurer. It takes an MD simulation and it looks at the pocket over the course of the whole simulation, and it calculates the volume and the shape of that pocket. And that makes it very easy then, once you know the volumes associated with each trajectory frame, to just pick out the ones that are the largest or the smallest or that have other features. So it can be downloaded from nbcr.ucsd.edu/POVME in all caps. And as I mentioned, there's also a copy there on your Ubuntu, NBCR Ubuntu image. So I thought, oh yeah. In fact, I forgot to mention, please interrupt me with questions. So yeah.
Yeah, I just wanted to ask you, can we use POVME to also find chemical sites?
So we're just working on a review where we talk about that very thing. Yeah. So there's an associated helper program called POVME Pocket ID, and you can use that to identify potential pockets, and could possibly be an allosteric pocket. And then, yeah, you could monitor the volume of that allosteric pocket as well over the course of an MD simulation and see, does it occasionally open up really widely on the course of a simulation, maybe be druggable in that conformation. So yeah, that would definitely be a good application. Any other questions? Please do interrupt because my lecture runs a bit short, so I welcome any questions you might have. So the first step in using POVME is you need to align your trajectory. You know, as in real life, there's Brownian motion, especially when you're simulating a small protein. It'll bounce all over the place. And POVME expects the pocket you're measuring to always be located in the same three-dimensional space. And you know, of course, the local motions of the pocket will change, but you need to remove any of the translation or rotation that's acting on the whole protein. There's many programs that can align trajectories like that. I really like Visual Molecular Dynamics. It's from Klaus Schulten's group, and it's a free program that can be easily found on the internet. So step 2 is to save the aligned trajectory in the PDB format. So most of the MD engines save it in a sort of binary or compressed format, but POVME needs this text-based PDB format. Future versions might be compatible with binary file formats, but fortunately, the VMD can also convert binary formats into the PDB, even a multi-frame PDB format, so you can really get the whole trajectory in a PDB. So the same program can be used to align and then just save as a PDB. So the third step is to define what I call a pocket inclusion region. So this is a region in three-dimensional space that's defined by a series of spheres and boxes, and it needs to encompass all of the pocket conformations of the entire, over the course of the entire trajectory. So it's good to kind of make it a little bit bigger than it really needs to be. And then after you've defined that region, it tells POVME which pocket you're interested in. Oh, I'm sorry, I'm getting ahead of myself here. I just wanted to, so I'm not going to go through all of the POVME commands. That'll get tedious, but you can see that online. There's a number of tutorials. Just give you a feel for how it's done. POVME has this POVME input file where you first give a command and then you give parameters. So to define that inclusion region I was just talking about, you could do points, inclusion sphere, and then the center, x, y, z coordinates of the sphere and the radius. And then points inclusion box is the center of the box, and then the x, y, z dimensions of the box. And so all of POVME's options have commands like this that you put in this input file that POVME reads. So the next option, and this is an optional feature, you can define what I call an exclusion region. So sometimes in the process of making an inclusion region, you want to make it big to encompass the whole pocket. But there might be a certain portion of the protein that you really want to say, this is not the pocket. It'll never be the pocket. And you could try to adjust your inclusion region just right, but sometimes it's easier just to say exclude this region of 3D space from being any kind of a pocket. And it's also comprised of spheres and rectangles, or boxes rather. Step 5 is then to fill that region with space. So it fills the inclusion region with points. Sorry, to fill it with points. And then it looks at each of those points and it removes any point that's also in the exclusion region. So fills the inclusion region and then removes points that are in the exclusion region. It is much easier just to show you. So if we have this defined as the inclusion region and this defined as the exclusion region, then this is the space that will be filled with these points. And the points are equidistant, so they each occupy the same volume. I think the default is 1 angstrom. And that's how then the volume of the pocket will ultimately be calculated. So the next step, any questions so far? Oh, nice.
So what's a good example for biologically for using exclusion?
So one good example would be if you have one pocket right here and there's another pocket right adjacent to it, and you really want to just look at the primary pocket, you want to make sure you're never getting the other pocket, then you might put an exclusion region just around the other pocket. And you could also imagine a scenario where you could pick your inclusion region just right so it never hit the other pocket, but sometimes it's easier just to exclude it. So does that make sense?
Yeah, thank you.
Any other questions? Nice. So the next step then is to hollow out these points. So POVME next takes a look at all of the points that are in that inclusion region and removes any that come close to any receptor atom. And so what's left are predominantly points that are just inside the pocket. But what I want to call to your attention is that there are still a few isolated little islands of points out sort of in the middle of nowhere that are not connected to the main pocket. And these probably aren't even real pockets. You know, it's just, it happens to be there's one little place where the point can avoid protein atoms. So that's an issue that we'll address in just a moment. But for the most part, these points are now in the pocket. And also, just to emphasize that POVME can work with whole trajectories. So if you feed it a multi-PDB or multi-frame PDB file, it'll do the same thing separately for every single frame so that you can monitor it over the course of the simulation. So next option is to remove any points that come outside of a convex hole defined by the protein. So, you know, a lot of these features were added as we realized different tricky aspects of the software. And one thing I realized is that when you make this inclusion region big, sometimes it'll sort of protrude out into the solvent space. It's not really a pocket. And so you can calculate, the program will calculate this convex hole, and you can optionally say, remove any points that are outside of that hole. So if you really want to get it just right at the opening of the channel, that can be a useful way of doing it. And then, getting towards the end of the different POVME options, you can also optionally define what I call a non-contiguous seed region. So this is speaking to the problem of these little isolated islands of points in the middle of nowhere. So again, using spheres and boxes, you can define a smaller region of 3D space that's always right in the pocket. And then POVME will automatically remove any points that aren't contiguous with that small region. So here, I've put this green sphere as my seed region right in the center of where I know that pocket, at least part of the pocket's always going to be. And POVME will then remove any points that aren't directly connected to that smaller region. So here you can see that we don't have any more of those little islands of points floating in the middle of the protein. So it's kind of a way of filtering to get even more accurate results. All right, the POVME output is just a text file that includes the volumes for every frame, also includes what user options people use, because I always forget what I did, so I can go back and see. And then also for every PDB frame, it'll output a separate PDB file showing the points that are in that pocket. So you could even examine it on a frame-by-frame basis. And then the latest version of POVME also outputs this volumetric density map in the Data Explorer DX format. So what you can do is you can also view this in VMD as an isosurface, and then that will represent the region of the pocket that is open over as a percentage of the whole MD trajectory. So for example, if I chose an ISO value of 0.25, it would show me this is the part of the pocket that's not buried in a protein, that's actual pocket, for 25% of the simulation or more. So if you wanted to see the very, very most persistent part of the pocket, you might pick 0.8 or 0.9. That's the part of the pocket that's there 90% of the time. And so this is really good for finding cryptic side pockets. You can start out at 0.9 and then slowly go down, and you can say, oh, well, look, this little side pocket opens up in 25% of the trajectories. And so that's a useful feature we added in just the latest version. So let's talk then about NNScore, second part. So getting back to the two steps of docking, we have first predicting the pose and then using a scoring function to try to estimate the binding affinity of that pose. So NNScore then is a rescoring function. It takes the pose found through some other method. Maybe you docked with AutoDock first, or maybe you have a crystallographic structure, and then it reevaluates that docked pose to try to come up with a better estimate of whether or not it's a good binder or a bad binder. So it's a docking rescoring function. And it's a novel approach in that it's based on neural networks. So what's a neural network? It's an attempt to model, albeit totally inadequately, sort of the functioning of the brain. In the brain you have different cell bodies. There's a certain architecture or configuration of the neurons, and they're connected through axons. And then also there's the strength of those connections. And those two things, architecture and strength, of course this is a ridiculously simplistic description, but those two factors can contribute to how these biological networks behave. And so the computational version is sort of inspired by that. There's a certain architecture. Basically these neurodes, or artificial neurons, are arranged in layers, and then there's fixed connections. Well, there's some methods where the connections are even variable, but for our purposes there's fixed connections, and you encode on the input layer, you encode information about the system you want to analyze. So an example from humans would be the retina, where you want to analyze information about the world around you. Light comes into your eye, hits your cones and rods and whatnot, and encodes that image on that input layer. Then the information goes, sort of percolates down through the network through optional hidden layers. And you can have, here I'm just showing one, but you can have multiple hidden layers. And then finally, it's modified by the weights of these different connections, and the analysis is encoded on that output layer. So again, this is a very simplistic comparison, but the output layer might be your occipital lobe, let's say. It's encoded on your retina, it goes through your optic chiasm and whatnot. And then finally in your occipital lobe, there's some level of understanding of the input or categorization. So in the context of trying to teach these networks to predict binding affinity, first on the input layer, we encode structural information about binding, and I'll describe that more in just a bit. Then we let that information sort of percolate down, and initially the connections are just randomly chosen, and so there's no meaningful output. It's just total rubbish. But what we do is we then systematically adjust the strengths of these connections until the networks start reproducing more or less accurately the experimental binding affinities. And so this is how we're training the networks, how they're learning to predict binding affinities just from characteristics of of the binding event itself. So how do we encode binding on that input layer? Well, there's a number of factors that go into it, minor details that if you're interested, you can look at the paper. But mostly, they're just dependent on pairs of juxtaposed atoms. So, how often does an atom on the receptor, carbon, say, come within 3 angstroms of an atom on the ligand? And so we tally up all those different kinds of atom pairs based on atom type, and then that's what goes on to the input layer of these networks. So, for example, let's say that there's a carbon atom on the ligand that comes within 3 angstroms of a carbon atom on the receptor. Well, then we're going to add that to the CC bin. Or if there's a hydrogen on the ligand that comes close to a nitrogen on the receptor, that'll be added to the HN bin. So you just get this big tally of these different pairs of atoms. Does that make sense? Any questions about that? And so ultimately, when we considered all the different atom types, we didn't do it just based on element. We looked at things like, well, it's actually the autodoc atom type, so I'll probably discuss that this morning. When we considered all that sort of thing, it turns out that we had 194 different ways of characterizing that binding event. And so our neural networks had 194 input neurodes on that initial layer. We did a lot of experimentation, and we found that a single hidden layer of 5 neurodes gave good performance. And it's going to be exciting going forward with GPU-accelerated neural network training. Maybe more complex architectures could be even more accurate. So there's a lot of possible exciting developments coming in the future. Then on the output layer, there's 2 neurodes, and we trained it so that 1, 0 means that the experimentally measured KD value was less than 25 micromolar, and 0, 1 means that it's greater than 25 micromolar. So this is NNScore 1 I'm describing, which unfortunately is slightly better performing than NNScore 2, but it depends on what receptor you pick. But anyway, NNScore 1 is a binary classifier. It'll just tell you whether it's a good or bad binder. So what we needed then to train these networks is we needed a vast database of crystal structures of protein-ligand complexes with associated experimentally measured KD values, so we could tell the neural networks what the right answer was, right, essentially. And fortunately, several people have put together some great databases like this, Binding MOAD and PDB Bind, just thousands of crystal structures. They're adding new crystal structures to it every year, but then also the associated binding affinities of the crystallized ligands and protein. So we took that data and we compiled a database of 4,141 protein-ligand complexes. 2,710 came from those 2 databases with known binding affinities. But then we noticed there's a certain bias in the literature because nobody publishes their lousy inhibitors. But it's important that the networks know what a lousy inhibitor looks like. So what we then did is we took 1,431 compounds from the National Cancer Institute. And this was the only time in my whole career when I was looking for examples of bad docking. Usually I'm trying to find the best one. But here I tried to intentionally find poorly docked compounds to serve as bad examples for the neural network. So in total, there were about 4,000 between the two of these. And we ultimately trained 1,000 different networks. We divided the database into training sets and testing sets. We trained them on the larger training set. But then when we went to evaluate how well they'd learned to predict binding, we looked at it on the testing set, which was comprised of examples that they'd never seen before, never been trained on before. And we found that when we judged molecular recognition by consensus, so not just asking one neural network, is this a good binder, but kind of what's the consensus of the top performing neural networks, that gave us much better results. So in that sense, it's kind of like democracy. If you put all the power in one person's hands, they tend to make bad decisions. But you'd like to think that if everybody gets together and we all try to decide what the right answer is, we'll have a better chance of getting the right one. And I certainly hope that happens in the coming election. But in the case of neural networks, I know it's true to do it by consensus because I actually tested it. In fact, we have a 90% accuracy on the testing set for crystallographic structures. Now, with docking, you introduce the additional inaccuracy of do I even have the right pose that I'm evaluating? But at least on the crystallographic structures, we were able to categorize with 90% accuracy. So there's been some subsequent developments since we initially came out with NNScore. One recent study examined 40 distinct protein targets from the DUD set, the old DUD set, and found that on average NNScore performed comparably to GLIDE, which is it depends on which receptor it is. Some receptors GLIDE's better. That's true of all scoring functions. But given that GLIDE costs thousands of dollars a year, and given that these neural network scoring functions are still in their infancy, that was a promising start. And NNScore was also used by others to identify a compound that was effective against MRSA in an in vivo phenotypic screen. So this was in a mouse model. So I haven't cured any people yet, but lab rats love me. I'm like a hero to them. And then it's also been used in a number of other projects. I'm biased in favor of this last one because we just published it, but this last one was published by our group, and we found some nanomolar inhibitors of the estrogen receptor, so using NNScore and other techniques. So there's a certain history of, it's a new scoring function, but there's a certain history there. I've also had 2 companies contact me, tell me it's being used in industry. I hope that there's others too, but I imagine not everybody contacts me. But anyway, one of them, Chemolytics, said, we did a comparison of GLIDE and VENA, and using the neural network rescoring, we clearly outperformed GLIDE. That was true for this particular receptor. Wouldn't be true for others, but still. Our clients now have a hard time getting, our clients have had a hard time getting decent hits for their targets. Now I think we have a path forward, at least to start screening and doing medchem. So it kind of warms your heart when you hear that your baby's gone out in the world and is doing well.